Hello everyone, my name is Sakshi Bhoir and my group members are Sapna Bhoir and Kajal Bhoir. We are studying in final year B Pharmacy in Shivajirao Jondhle College of Pharmacy, Asanga. Today we are going to explain about the topic tannins, guided by Professor Manisha Vite. So let's start with the topic. Tannin. It is derived from the French word tannin, which means tannin substance. Tannins are complex organic non-nitrogenous plant products which generally have astringent properties. The term tannin was first used by Seguin in 1796. Tannins belong to the phenolic class of secondary metabolites and are formed through shikinic acid pathway. Classification of tannins. On the basis of Goldbeater skin test, the tannins are classified as true tannins and pseudotannins. True tannins are the ones which give positive tannin test and pseudotannins are the ones which fail to give the positive test. On the basis of hydrolytic reaction, they are classified as hydrolyzable tannins and condensed tannins. Hydrolyzable tannins are the ones which are easily hydrolyzed by the mineral acids or tannase enzyme. Examples are Amla, Bhoir, myrobalan, and Arjuna. Condensed tannins. These are the tannins that are not easily hydrolyzed. Example Ashoka, pale Catechu, black Catechu, and pterocarpus. Characteristics of tannins. Tannins are colloidal solutions with water. They are non-crystalline substances. They are soluble in water, sparingly soluble in ethyl acetate, and insoluble in organic solvents except acetone. Their molecular weight ranges from 500 to more than 20,000. Oligomeric compounds with multiple structure units with free phenolic groups. They can bind with proteins and form insoluble or soluble tannin protein complexes. Chemical test for tannins. First, Goldbeater's skin test. In this, we take a piece of Goldbeater skin previously soaked in 2% hydrochloric acid and then washed with distilled water. Place this in a solution of tannin for 5 minutes. Wash with distilled water and transfer to 1% ferrous sulfate solution. The change in color of the Goldbeater skin to brown or black indicates the presence of tannins. Hydrolyzable and condensed tannins both gives the positive Goldbeater's test, whereas pseudotannins show a very little color or negative test. Second is phenazone test. To 5 ml of aqueous solution of tannin containing drug, add 0.5 gram of sodium acid phosphate. Then warm the solution. Then cool and filter it. Then add 2% phenazone solution to the filtrate. The tannins are precipitated as bulky colored precipitate. Third is gelatin test. Take 1% gelatin solution plus little 10% sodium chloride. Then add 1% solution of tannins. The precipitation of gelatin from solution occurs. Now let's start with the hydrolyzable tannins. First drug is Amla. Synonyms are Imblica and Indian gooseberry. Biological source is Amla consists of fresh as well as dried fruits of Imblica officinalis gourd belonging to the family Euphorbiaceae. Geographical sources are deciduous forest of India, Sri Lanka and Myanmar. Cultivation and collection. Soil. Light heavy soil and loamy soils are required. Rainfall required is 630 to 800 mm. Drip irrigation is used. Tolerate freezing temperature and high temperature up to 46 degrees Celsius is needed. Propagation is done by seed planting. Harvesting is done in the month of March to May. Yield is 50 to 70 kg per plant and big size fruits are collected. Morphology. Color. Green color changing to light yellow or brick red when matured. Odor. None. Taste is sour and astringent. Shape. The fruits are depressed and globose. Size is 1.5 to 2.5 cm in diameter. Extra features. Fruits are fleshy, obscurely four-lobed with 6 trigonous seeds. They are very hard and smooth in appearance. Microscopy. Epidermis contains a thick cuticle and 2 to 4 layers of hypodermis. The cells in hypodermis are tangentially elongated, thick-walled and smaller in dimension than epidermal cells. Mesocarp. It consists of thin-walled isodiametric parenchymatous cells, several collateral fibrovascular bundles, and also aggregates of numerous irregular silica crystals are present. Xylem is composed of tracheal elements, fiber trachees, and xylem fibers. Tracheal elements show reticulate scleriform and spiral thickenings. Chemical constituents. Amla is highly nutritious and its main chemical constituent is vitamin C. Minerals and amino acids are also present. The pulpy portion of the fruit contains tannins, which includes gallic acid, ellagic acid, and phylembin. Sugar, gum, albumin, and crude cellulose are also present. Chemical test. First one is to alcoholic or aqueous extract of the drug, ferric chloride solution is added which gives blue color. Second, to aqueous extract add gelatin and sodium chloride, then the milky white color appears. Third, to aqueous extract of amla, lead acetate is added, then precipitate formed is removed by filtration. To the filtrate, we add solution of 2:6 dichlorophenol and indophenol, then the color disappears. Uses. Amla is used in hemorrhage, diarrhea, diabetes, and dysentery. It is also used in disorders associated with the digestive system and also prescribed in treatment of jaundice and cough. It has antioxidant, antibacterial, antifungal, and antiviral activities. Marketed products. The marketed products are Jivani Malt from Chirayu Pharma, Triphala Churna from Jondhle, and Chawanprash from Dabur. Next drug will be explained by Kajal Bhoir.
Next is Arjuna. Synonym of Arjuna is Arjun bark. Biological source. Arjuna consists of dried stem bark of the plant known as Terminalia arjuna belonging to family Combretaceae. Geographical source. It is mainly found in Indian peninsula, commonly in Chota Nagpur. Then the cultivation and collection. It required the loamy soil. Climate should be subtropical or tropical. Rainfall from 1000 to 1500 mm. Temperature should be 20 to 30 degrees Celsius. Propagation done by the seeds. Fertilizers which are used as FYM and the NPK. Pesticides, spraying of biopesticides. Then the collection is done by the collected bark is dried in a well ventilated shed and stored in boxes. Then the morphology. Color of the outer side as well as inner side of bark is grayish brown. It don't have any odor. Taste is astringent. Shape is flat. Size, the pieces of various sizes about 15 by 10 by 1 centimeter. Then the extra features. As Arjuna is collected from the old trees, the cork gets removed due to the exfoliation. The appearance of transversely cut surface is dark brown with a characteristic grayish shining patches. Then the microscopy. Here we can see the tears of the Arjuna. The uppermost part of the tears is known as the cork. Also we can see here the pericyclic fibers are present. Crystals, that is the calcium oxalate crystals are present. Cork uniformly arrange several layers of small tangentially elongated cells. Cortex, composed of thin-walled, more or less brick-shaped parenchymatous cells containing cluster crystals of calcium oxalates. Sclerenchymatous pericyclic fibers are also present. Secondary phloem consists of phloem parenchyma composed of thin-walled polygonal cells with wavy walls containing cluster crystals of calcium oxalate and the pigmented cells. Narrow and almost straight medullary rays are also present. Then the chemical constituents. It mainly contains the then the chemical constituents. It contains the 20 to 24% of tannins, flavonoids such as the arginone, and the bacillin, triterpenoids compounds, arginine, arbugenin, arjuglucoside 1 and 2, and the terminoic acid. Also contains the arginoglucoside 3. Here we can see the structure of the arginalone and the arginoglycoside 3. Then the chemical test. Ethereal extract of Arjuna shows a pinkish fluorescence under ultraviolet light. Then the uses of Arjuna. It is used as a diuretic and astringent, used in treatment of various heart diseases. Also, the bark was extensively used in the past by the local tanneries for tanning animal hides. Lots of marketed preparations are available from them. Here we can see the some. First one is the Mentat, which is from the Himalaya Drug Company. Then the Arjun Grita or Arjun Churna, which is from the Baidyanath Company. Then the Madhu Doshantak, which is from the Jamuna Pharma. Then the Ashoka. Synonym of Ashoka is the Ashoka bark. Biological source. Ashoka consists of dried stem bark of the plant Sarava indica belonging to family Leguminosae. Geographical source. It is mainly found in Malaysia, Indonesia, Sri Lanka and India. Cultivation and collection. It required the loamy, sandy loamy or clay loamy soil. Climate should be moist tropical areas. Rainfall is well distributed. Propagation done by the seeds. Seedling in a nursery in March. Collection done by the bark is dried in the shade, packed and stored in the containers. Then the morphology. Color. Outer dark brown or almost black with the warty. It don't have any odor. Taste is astringent. Shape. Form of channels of various sizes. Size up to 50 cm length and 1 cm in thickness. Then the microscopy. Here we can see the TS of the Ashoka. The uppermost part of the TS is known as the cork. Also present the scleroids, pericyclic fibers, phloem fibers, medullary rays and the crystals of the outermost layer are a few layers of phloem and phloem. Phloem contains stone cells in the form of distinct rings and also large stands. Phloem fibers are present in small groups, crystal fibers and the funnel-shaped uniseriated medullary rays in the inner bark. Starch is also present to small extent. Then the chemical constituents. It contains the mainly leucopelargonium and the leuconidine, 24-methylcholesterol 5 in triol. Then the chemical test. Powdered bark when treated with the saturated picric acid solution remains brown for 10 minutes and then slowly turns to orange yellow. Then the powder bark gives a deep chocolate color with a 5% KOH solution. Then the uses. It is used as a uterine tonic, as a sedative, suggested in all cases of uterine bleeding, stimulant effect on the endometrium and the ovarian tissue, also useful in the menorrhagia. Then the marketed products. The Mensa from the Lupin Herbal Laboratory, then the Femiplex from the Charak Pharma, then the Ashoka Rishta from the Baidyanath.
Hello, I am Sapna. I will explain the next drug, that is black Catechu. Synonym: Kach, black catechu and Katha. Biological source: black catechu is a dried aqueous extract prepared from heartwood of Acacia catechu wildano, belonging to family Leguminaceae. Geographical source is Madhya Pradesh, Maharashtra, Gujarat, Rajasthan, Bihar and Tamil Nadu. Cultivation and collection. Soil is a well-drained and neutral soil. Climate required for black catechu is tropical and subtropical. Rainfall is about 500 to 2000 mm. Temperature is 32 to 39 degrees Celsius. Propagation by seeds. Fertilizers used for black catechu are green manure and FYM. Collection. The heartwood is collected and cut into the pieces and dry in a shadow. Then morphology. Color is black or brownish black mass. Odor, odorless. Taste, astringent and subsequently sweet taste. Size, irregular mass. Extra features of black catechu are outer surface is firm and brittle. When broken, the fracture surface appears glassy with small cavities. This is the image of the black catechu. Microscopy. Heartwood shows the numerous uniseriated and biseriated medullary rays with vesicles occurring in small groups of 2 or 4. Xylem fibers have a narrow lumen and xylem parenchyma is usually predominantly paratracheal, forming a sheath around the vessels. Wood consists of crustal fibers having prismatic crystal of calcium oxalate. A few tracheids with scleriform thickening and some cell including the vessels are also present. Then chemical constituent 2 to 12% of catechin, which is the main chemical constituent of black catechu. The structure of catechin is shown on the slide. 25 to 33% of phlebotannin, then catechu, tannic acid, epicatechin, acatechin, D-L-acacatechin, acacatechin and D-isoacacatechin. These are the chemical constituents are present in the black catechu. Also present the catechinic acid, quercetin and quercetin. Then chemical test used for identification of black catechu. Black catechu gives the pink or red color with vanillin and HCl because of presence of catechin. The next test is catechin when treated with HCl produces the fluoroglycerol, which burns along with lignin to give the purple or magenta color. Now next the uses of black catechu. Black catechu is used as an astringent, also cures the trouble of mouth, diseases of throat and diarrhea. It also increases the appetite. Black catechu is used in a dyeing industry. Kach is used to dyeing the fabric brown or black. It's also used in a calico printing. Next, the marketed products of black catechu. First is Cofflet lozenges, which is manufactured by Himalaya Drug Company, and next is Gumtone, which is manufacturing by Charak Pharma Private Limited. Says Textbook of Pharmacognosy and Phytochemistry, author Biren Shah and A.K. Seth. Some links from Google. We are thankful to our President Honorable Dr. Shivajirao H. Jondhle, Sir, Secretary Honorable Dr. Geeta Khare Ma'am, and Principal Dr. Nitin Bhoir Sir for encouragement and guidance for efficient use of social media to spread knowledge. Please like and subscribe our channel for such informative videos. Thank you guys.